In this video, we will demonstrate different ways you can view gene expression data on the GTEx portal, whether you have a single gene of interest, multiple genes of interest, or a tissue of interest. If you have a single gene of interest, you can enter the gene identifier in the expression menu or in the search box on the right side of the menu bar. We will search for the gene P10. Note that you can enter a gene symbol, a GenCode ID, or an ensemble ID. At this time, the GTEx portal is displaying release version 7 of the GTEx data. This release uses GenCode version 19. If you enter a gene identifier that is not in GenCode version 19, the GTEx portal will not find that gene. The gene page displays a violin plot of the expression of your selected gene by tissue. If you mouse over a tissue on the violin plot, you will see a pop-up dialog that displays the name of the selected tissue, the median TPM, and the number of samples for that tissue. The controls above the violin plot provide additional options for controlling the violin plot display. By default, the tissues are sorted alphabetically, but you can also sort by median TPM. The scale control allows you to choose between the vertical scale being linear or log. The tissue filter button allows you to select which tissues are displayed. The subset button allows you to view expression grouped by sex. If you have multiple genes of interest, you can use our MultiGene Query page. Enter a list of up to 50 gene identifiers. We will use the example link to enter a list of gene identifiers. As you can see, these identifiers include a mix of gene symbols and GenCode IDs. You can also select which tissues you want to see. We'll accept the default of all the tissues and hit the search button. This displays a heat map of expression of the selected genes across the GTEx tissues. By default, the rows and columns are clustered. The controls above the heat map allow you to download the image in SVG format, sort the tissues alphabetically, or to cluster the tissues. You can select a gene by clicking on the gene symbol to the right of the heat map. This will display violin plots of expression by tissue for that gene beneath the heat map. You can select additional genes by clicking on the gene symbol. Clicking on a gene that is already selected will deselect that gene. If you have a tissue of interest, you can use our Top Expressed Genes page. We will navigate to this page using the expression menu. We will then select a tissue, adrenal gland. This displays a heat map containing the 50 genes most highly expressed in adrenal gland, showing their expression across all GTEx tissues. The controls above the heat map allow us to perform a number of actions. We can download the heat map in SVG format. By default, mitochondrial genes are excluded from the top 50 expressed genes. But if we select this icon, we can choose to display mitochondrial genes. Or we can choose to exclude them. By default, tissues are clustered, but we can choose to sort the tissues alphabetically instead. Similar to the MultiGene Query page, we can click on a gene symbol to select that gene and view expression across tissues in a set of violin plots beneath the heat map. You can select additional genes by clicking on the gene symbol, and this displays additional violin plots. Clicking on a gene that is already selected will deselect that gene and remove it from the violin plots.